Hi everyone, this is your biology class. I'm your teacher C. Madreja. Student, in this video we are going to discuss the development of monocot embryo from the single cell zygote. As we all know, single cell zygote is the product of syngamy or fertilization, in which one male gamete fuses with the egg cell and the zygote is produced. So in zygote there is diploid number of chromosomes are there. What happens in case of zygote? At the time of development of embryo, transverse division takes place and three cell structure is produced. First transfer division takes place, two cells are produced, and then out of two cell only one divides. So three cell structure is produced after transfer division in zygote. The cell which is present towards the micropillar end is known as the basal cell. The cell which is present towards the chalazial end is known as the terminal cell, and the middle cell is known as the middle cell. Now we are discussing the fate of all these three cells. Basal cell simply enlarge in size. It do not participate in the formation of any structure belonging to embryo. It simply enlarge in size and help in the absorption of nourishment. बेसल सेल में किसी तरह का कोई डिवीज़न नहीं होता है, ये सिंपली साइज में इंक्रीज कर जाती है और नरिशमेंट गेन करने में हेल्प करती है. द टर्मिनल सेल गिव राइज only one cotyledon. In case of monocot embryo, only single cotyledon is present and we called it as scutellum, which is produced from the terminal cell on further development. All the parts of the embryo are produced by the middle cell. In the middle cell, the division takes place in all the planes and group of cells are produced which later on give rise to other parts, all parts of the embryo like plumule, radicle, epicotyle, hypocotyle. So this is the fate of all these three cells. I am going to draw the structure now. From the basal cell, no further division takes place. It's only enlarge in size and help in the nourishment or absorption of the nourishment. This is the basal cell, which simply enlarge in size. In middle cell, division takes place in all the planes, so it gives rise to mass of cells. These are the group of cells which are produced by division into the middle cell. From the terminal cell, single cotyledons are produced. It divides in all planes and group of cells are also produced from the terminal cell, which later on give rise to cotyledon. Now this structure is reorganized and give rise to the following shape. Basal cell is present here, which simply enlarge in size. These group of cells which are produced from the middle cell, they differentiated and start producing plumule-like structure. On the other end of this embryonal axis, there is present the radicle-like structure. We use the other terms for plumule and radicle in case of monocot embryo. We use these terms later on. Between the plumule and radicle, where cotyledon is attached with this embryonal axis, is known as the cotyledonary node. The place between plumule and cotyledonary node is known as the epicotyle. This is epicotyle region. And the area between the radicle and cotyledonary node is known as the hypocotyle region. So this is how group of cells which are produced from the middle cell, they differentiated to give rise the different parts of the embryonal axis. With the help of terminal cell, single cotyledon is produced, which we called is as scutellum in case of monocot. Now there is modification in this structure takes place. The structure like this is developed. This is the embryonal axis. This is the plumule part. This is the radical part. This is cotyledonary node. through which single cotyledon or scutellum is found attached. In case of monocot, embryo radicle is covered with the help of a protective covering that is known as the coleoptile. So we called it as coleoptile, which represents the radicle. And on the other embryonal axis, plumule is also covered by protective covering of cells that is known as the coleorhiza. So we use in case of monocot term, at the place of radicle we called it as coleoptile. At the place of plumule we called it as coleoptile. At the place of radicle we use the term coleorhiza. In between them it is quaternary node through which single cotyledon or scutellum is found attached. Scutellum or cotyledon in this case is thin and papery. The food is not stored into the cotyledon in case of monocot embryo, and we call the cotyledon as scutellum. This structure is present inside the seed. Now I am going to draw the structure of the maize grain. Inside this we form this embryonal axis. The shape of the monocot seed in case of maize grain is like this. It is triangular in outline. It is divided into two compartments with the help of epithelial layer. This is the epithelial layer which divides the inside of the seed coat into two chambers. One is the upper chamber which is bigger in size, and the lower one is the smaller chamber in which embryo is placed. This is the embryo. This part showing coleoptile. On the other end it is coleorhiza through which attached the single cotyledon, that is scutellum. Scutellum is papery and thin. No reserve food is present into the scutellum. The upper chamber of the seed of maize grain consists of the endosperm. The outer layer of the endosperm is known as the aileron layer. It is made up of protein and we called it as proteinaceous endosperm. And in the center, starch is deposited into the inside of the seed and represents the starchy endosperm. This is the structure of the maize grain. What happens as the scutellum or cotyledon is thin and papery, there is no reserve food. The function of cotyledon in case of dicot embryo, it supplies nourishment to the developing embryo. But in case of monocot, the scutellum or cotyledon, no reserve food material is there, it is papery. Then what is the function of this? When the endosperm gets digested with the help of phytohormones, then it becomes digested food, and this digested food is supplying to the embryonal axis, that is coleoptile and coleorhiza, for the growth of the embryo. Then the food reserve present into the endosperm after digestion supply first to the cotyledon, and then with the help of cotyledon or scutellum they passes to the embryonal axis. I think the concept of development of embryo in case of monocot is very much clear to you all. एक बार quick revision लेते हैं. What happens in this case? First, zygote divides into by transverse division, and 3 cells are produced. Towards the micropillar and basal cell produced. In the middle cell is there, and towards the chalazial and terminal cell is produced. With the help of terminal cell, only one cotyledon is produced. Middle cell forms all the several parts of the embryo and divides in all plane. Basal cell, there is no division takes place, only it enlarges in size and help in the absorption of the nourishment. After further development in all the 3 cells, this type of structure we obtain. This is the basal cells, generally enlarges in size and help in the nourishment of the embryo. Middle cell divides and redivides in all planes, give rise to mass of cells. Terminal cell give rise to single cotyledon. So such kind of structure is produced by these cells it is the basal swollen cell. This is the embryonal axis, which is produced by this mass of cells produced by middle cell. This is the embryonal axis. On one end, plumule like structure is there. On the other end, radicle is differentiated. This is single cotyledon scutellum attached with the point that is known as cotyledonary node. The place or point present between the cotyledonary node and plumule is known as the epicotyle, and the area present between radicle and cotyledonary node is known as the hypocotyle. A little suspensor is also there, which is present here. Now this structure becomes like this. This is the embryonal axis. On one end, radicle is covered by the protective sheath, and in case of monocot we called it as coleoptile. This is plumule, which is known as the coleoptile. On the other hand, it is radicle covered by the protective covering. We called it as coleorhiza. Cotyledon is attached with the cotyledonary node. This is cotyledonary node. In between cotyledonary node and coleoptile, this is known as the epicotyle. And the region between cotyledonary node and the coleorhiza is known as the hypocotyle. This is single cotyledon which is attached with the embryonal axis. We called it as scutellum. This structure is placed inside the seed coat, is present inside the seed coat. This is the diagram of monocot seed, that is maize grain. Two compartments are there. The lower compartment, in which embryonal axis is present. Dono compartments are separated with the help of epithelial layer. Embryonal axis, that is coleoptile and coleorhiza, and single cotyledon are present into the smaller chamber, and the bigger chamber there is present the endosperm. The outer endosperm is proteinaceous and is known as the aileron layer, and in the center starchy endosperm is deposited. On digestion, food material in the endosperm comes in dilution or solution form and passes to the cotyledon, and then from the cotyledon it passes to the embryonal axis. This is all about in this video. I think the concept is very much clear to you all. Thank you so much.